Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about different types of PETS clamp techniques. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. PETS clamp technique is also called PETS clamp recording. In this technique, a micropipette is used as a patch which is clamped or tightly attached to the cell surface. Hence, the name is patch clamp technique. There are three main types of patch clamp techniques. First one is whole cell patch clamp technique, in which after the formation of a tight seal between the cell membrane and a micropipette, the membrane patch which is present within the pipette is disrupted by briefly applying strong suction. This disruption creates a passage between micropipette and the cytoplasm. Such an arrangement makes it possible for the determination of electrical potential and current from the entire cell. Hence, this technique is called whole cell patch clamp technique. It also allows for the diffusional exchange of ions and molecules between the pipette and the cytoplasm. Whole cell patch clamp technique can be performed in two modes. First one is current clamp mode and the second one is voltage clamp mode. In current clamp mode, the current is kept constant and the changes in membrane potential is measured. In this mode, an electrode is used to inject a known current into the cell and another electrode sends the potential. Second mode is voltage clamp mode, in which the voltage is kept constant and the resulting current is recorded. In this mode, the potential within a cell is compared with the known voltage supplied by the experimenter, and an amplifier is used to supply whatever current is required to keep the potential at the specified voltage. An alternative variant of whole cell patch clamp configuration is perforated patch clamp technique. In order to explain the perforated patch clamp mode, we have taken the example of a nerve cell or neuron. This is a nerve cell in which the cell membrane contains numerous ion channels. And this is a micropipette which has been tightly attached to its surface. In case of perforated patch clamp mode, the patch membrane is not ruptured by using suction. Instead, the membrane is perforated by using small amount of antifungal and antibiotic agents in the electrode solution. These agents are used as perforants. Antibiotics, which are generally included in the electrode solution, diffuse to the membrane surface where they form many small pores in the patch membrane. Certain antibiotics such as nystatin, amphotericin and gramicidin are used as perforants. As already mentioned, in whole cell patch clamp configuration, the patch membrane is ruptured by using suction. So the whole cell patch is like an open door where there is complete exchange of molecules between the pipette solution and the cytoplasm. Unlike whole cell patch, Perforated patch is like a screen door that allows the exchange of certain small molecules between the pipette solution and the cytoplasm. The small pores in the patch membrane allow the diffusion of certain monovalent ions, enabling electrical access to the cell interior and preventing dialysis of large molecules and ions. Second type of patch clamp technique is inside out patch clamp, in which after the formation of the tight seal, the micropipette is retracted in such a way that two pieces of membrane remain attached to the pipette without disrupting the seal. The intracellular surface of these two membrane patches remain exposed. This technique is used to investigate single channel activity. Third type of patch clamp technique is outside out patch clamp. In this technique, after the formation of a tight seal between the pipette and the cell membrane, the patch membrane is disrupted by briefly applying strong suction. After disrupting the patch membrane, the pipette is removed in such a way that two membrane pieces remain attached to the pipette without disrupting the seal. When these two ends of the membrane patches are joined, a vesicular structure is formed. The extracellular surface of this vesicular structure remain exposed. This technique is used to study how channel activity is influenced by extracellular chemical signal such as neurotransmitter. Friends, this is all about different types of patch clamp techniques. Goodbye and good night.